The skin, or the integument, is the largest organ of your body, and its condition is able to communicate things such as health, age, sex, beauty, and it helps to define our self-image. But what about its physiological functions? The skin is obviously involved in protection, and especially from physical injury, chemicals, toxins, microbes, even excessive heat and cold. It's capable of regenerating itself, and it's also selectively permeable to certain chemicals, hence the effectiveness of things such as an estrogen patch or a nicotine patch. The integument also helps to prevent water loss. It is considered to be water resistant, so water can't exit or enter, the main exception being sweat glands. And this is one of the reasons that burn victims are in danger of dehydrating. Now, interstitial fluids may slowly diffuse to the surface of the epidermis and ultimately evaporate. In fact, you lose about a half a liter or 2 cups this way every day. And also from the respiratory system and from talking, those things also contribute to this water loss. And this water loss across the skin is referred to as transepidermal water loss. The skin assists with temperature regulation, so blood flow patterns. So when you're hot, vessels can dilate to dissipate excess heat. Sweat glands release sweat. When you're cold, vessels constrict and can shunt blood to the core. Your skin also assists with metabolic regulations. So vitamin D is synthesized by some of your epidermal cells when you're exposed to UV radiation. And this vitamin D is then important for the absorption of calcium and phosphorus from ingested food in the small intestine. In fact, you require about 15 minutes a day of unprotected exposure to sunlight to generate the amount of vitamin D that your body requires on a daily basis. The integument also contributes to the body's immune defense. You have epidermal dendritic cells or phagocytic cells that can help initiate an immune response against pathogens that invade through the epidermis. And the integument also helps, and the cells in the integument can also fight against epidermal cancer cells. Sensory reception. You have a variety of sensory receptors in your skin that are responsible for sensing things such as heat, cold, touch, pressure, texture, or vibration. And then also excretion through secretion. We've already talked about how water can do this, but sweat may carry waste products such as salt and urea, and you also have sebaceous glands that secrete oils. The skin is comprised of 2 layers. The epidermis is the superficial layer, and then below this, deeper to this, is where we're going to have the dermis. Now you have a 3rd layer that is actually deep to the dermis, but this is not considered to be part of the integument. So please only consider the fact that the integument consists of the epidermis and the dermis. There are several types of connective tissue that we're going to find in here, in addition to things such as the muscular tissue and the nervous tissue as well. The hypodermis consists of primarily adipose connective tissue, though also it contains some areolar connective tissue. The dermis contains areolar connective tissue as well as dense irregular connective tissue. And then in the epidermis is where we're going to find epithelial tissue. Now this is the one major type of tissue that we haven't talked about yet in this class. We've covered nervous tissue, muscle tissue, and connective tissue, but now it's time for us to move on to epithelial tissue. All of your organs are lined on the outside and on the inside by epithelial tissue. Some of these linings are also referred to as the endothelium, and the endothelium lines hollow organs and body cavities that are not connected to the exterior part of the body. Epithelial tissue provides protection from physical, chemical, and biological wear and tear. So what are some of the characteristics of this tissue? One is cellularity. This tissue is composed almost entirely of cells with little extracellular matrix. The cells are bound together by several types of intercellular junctions. These cells exhibit polarity, so epithelial cells have a top surface and a bottom surface that we refer to as apical and basal. The apical surface is the one that's exposed to the open cavities, and the basal surface of the cell is where they attach to the underlying cells. The basal surface is attached to what's referred to as the basement membrane, which is an acellular structure that's produced by both epithelial tissue and underlying connective tissue cells. Epithelial tissue is avascular. It does not have any blood vessels in it. So because of this, the cells receive their nutrients solely by diffusion from the underlying tissues. Innervation. Epithelia are richly innervated in order to detect changes in the environment at the body or the region of the organs. And they have a high capacity for regeneration. So epithelial cells are frequently damaged or die because of these environments that they're exposed to, and so they're replaced as quickly as they're lost. This is showing how, a little illustration of the general structure of the epithelial tissue. So we can see how here we have the connective tissue that's underneath, and then just superficial to that is where we're going to find the basement membrane. And then above that, we're going to find our layer of epithelial cells. This happens to be an example that only contains a single layer of cells. There is a basal surface of these cells, and then there is also the apical surface. That basement membrane, as I mentioned, is responsible not only for some physical support, but also some anchorage and regulation as well, by regulating movement of materials between the epithelium and the connective tissue that's underneath. So epithelial tissue can be classified based on the number of layers that it has and also based on the shape of its cells. I gave a short little preview about this when we were talking about layers of blood vessels, but just as a quick little review here. If we can classify the tissue based on the layers as either being simple or stratified. So a simple epithelium consists of a single layer of cells, and all of the cells have an apical surface and a basal surface that's attached to the basement membrane. Stratified epithelium, on the other hand, contains two or more layers of cells, and some of it can contain many, many layers. Not all of the cells have an apical surface, and not all of the cells attach to the basement membrane. Only the deepest cells have a basal surface, and only the most superficial cells have an apical surface. In terms of their shape, we can classify epithelial cells as either squamous, cuboidal, or columnar. Squamous cells are flat and wide and can often be irregular in shape. Cuboidal cells are about the same size on all sides, and when this is in these cells, the nucleus tends to be very centrally located. And in columnar cells, they're taller than they are wide. The nucleus tends to be more oval-shaped in a columnar cell and is located a little bit more towards the basal region, even though that might not be completely obvious from this illustration. You can have a transitional epithelial cells as well. And we're going to find this in a couple different organs where these organs stretch. And so transitional epithelial cells are able to change their shape. So we'll find these in things such as the urinary bladder. They look squamous when they're stretched, but when they're unstretched, the apical surface looks a little more cuboidal. So now we can combine all these different layers with some of these different shapes to come up with the general categories of epithelial tissues. So if we're looking at squamous cells, we can have a simple squamous epithelium or a stratified squamous epithelium. If we're cuboidal, we can have a simple cuboidal or a stratified cuboidal. And then columnar, we can have simple columnar, stratified columnar, or what we refer to as pseudo-stratified. Pseudo means fake. And so you can see in these cells, it almost might look like they are going to be stratified because the nuclei are at different levels, and some of the cells are shaped irregularly. So some of them are wider at the apical surface and thinner at the basal surface, and some of them are the exact opposite. And so you're always going to find the nuclei in the wider part of the cells. However, if you look closely at them, they are just a single cell layer thick bit here. So let's start by looking at these squamous cells. Squamous actually means scales. And this is showing you an example of what a squamous epithelial tissue would look like. This is actually a light micrograph of the air sacs of the lungs. So again, when we have this simple squamous, the nucleus tends to bulge in the middle, and we're mostly going to find this tissue when we need diffusion or filtration to occur, because it's a lot easier for that to occur over a single layer of cells. Stratified epithelium, again, has many layers of squamous cells. And a lot of times the ones that are more basal will be cuboidal in shape. Because of these multiple layers, it helps to protect the underlying tissues, often from abrasion. And this happens to be a photograph from the vaginal lining. This is an example of some tissue that is considered to be non-keratinized. So the cells don't have keratin, which is a protein being produced in them. The non-keratinized cells are found in places like the oral cavity, the pharynx, esophagus, the vagina, and the anus. Keratinized stratified epithelial tissue does have keratin in it, and that's to provide some additional protection. And this is only characteristic of the epidermis of the skin. So anywhere you're finding in the body stratified squamous epithelial tissue, except for the epidermis, it is going to be non-keratinized. And this is something I want you to sit and practice saying this over and over and over again, because frequently, especially when we get to a latter part of the course, when we are looking at some of these internal organ systems, we are constantly going to be saying the term non-keratinized stratified squamous epithelium. This is an example here of keratinized stratified epithelium. This is the layer of the epidermis, and this provides essentially even more protection because of those proteins. Cuboidal, simple cuboidal and stratified cuboidal. I will say to start with that stratified cuboidal epithelial tissue is very rare. This is an example of some of the tubules of the kidney. I want you to kind of think about simple cuboidal cells as being found when we have to be concerned with absorption or secretion. These cuboidal cells, again, are approximately equal height and width. They have a centrally located nucleus, and they help to facilitate the processes of absorption of substances in fluid and also secretion of molecules across that apical surface. So this happens to be an example of a photograph of the kidney tubules, which have to be able to resorb nutrients, ions, and water that's being filtered from the blood plasma. You're also going to find these types of cells in ducts and secretory regions of the exocrine or sweat glands. And then, as I mentioned, stratified cuboidal epithelial tissue is quite rare. Mostly going to be found in salivary glands and also in the ducts of sweat glands. So then how about the columnar? You've noticed I've put little parentheses around these cilia structures that I mentioned because you can have simple columnar without cilia. And whether or not the cilia there are often going to be related to whether you need to move substances around at that apical surface. Stratified columnar, quite rare. Here's an example of some simple columnar epithelial cells that are in the small intestine. And as we can see here, these cells do not have cilia on them. If simple columnar epithelial cells are non-ciliated, they tend to be associated with absorption or secretions, for example, in the digestive tract. While if they're ciliated, those tend to assist with the secretion of mucus and also movement. So, for example, this image here happens to be from the uterine tubes. As I said a minute ago, the stratified columnar epithelial tissue is quite rare, and this happens when and can be found when you need a combination of protection and secretion. So this happens to be an example of a photograph from the parotid gland, and so we can see here approximately where the two shapes, the two layers of these columnar cells are. So then how about that pseudostratified columnar epithelium? Just as I mentioned, you need to practice saying over and over again non-keratinized stratified squamous epithelium. Practice saying over and over again pseudostratified ciliated columnar epithelium, because these are the two that we're going to find quite frequently, especially in the respiratory tract. These are involved with protection and secretion, but again, when we need to move that secretion, or especially if it's a mucus, that we need to move around. So again, let's look at some of this classification kind of in the similar family tree-like fashion that we used with connective tissue. So squamous, we can have simple squamous and stratified squamous, and the stratified can also be classified as keratinized or non-keratinized, and we're only going to find keratinized in the epidermis. Cuboidal, simple cuboidal, and the rare stratified cuboidal. And then columnar, with simple columnar, pseudostratified, and stratified. And when we're looking at simple columnar and pseudostratified columnar, we usually need to specify whether it is ciliated or non-ciliated. Now let's return to the integument. So we mentioned that the integument consisted of the epidermis and the dermis, and then below the integument is where we find the hypodermis. Now, epidermis is where we're going to find our epithelial tissue in multiple different layers, as you can even see in this illustration. So let's go ahead and look at these different layers. Why am I talking about alcohol here? Come, let's get some beer. This is a mnemonic device for remembering what these layers are of the epidermis, going from the apical surface all the way down to the basal surface. So C, L, G, S, and B. Come, let's get some beer. Let's go through these, but starting with the beer. At the very bottom, at that most basal surface, is where we're going to find the stratum basale. Stratum basale is a single layer of cuboidal epithelium. It contains keratinocytes, which produce keratin, and the keratin helps to protect and waterproof the skin. It also contains melanocytes, which produce the pigment melanin, and melanin is able to absorb UV light to prevent DNA damage. And you're also going to find tactile cells there that are sensitive to touch. So here, for example, we can find a tactile cell that's connected here to a nerve ending, and here's an example of a melanocyte. Superficial to the stratum basale is the stratum spinosum. Stratum spinosum is several layers thick, and it contains still living keratinocytes. The daughter cells are the keratinocytes from the stratum basale, and then as they go into the stratum spinosum, they start to differentiate into non-dividing and highly specialized keratinocytes. Here's where you're also going to find epidermal dendritic cells, such as you can see here right where the arrow is. And those are immune cells that help to fight infection. They're phagocytes that initiate this immune response. Superficial to the stratum spinosum is the stratum granulosum. This is usually 3 to 5 layers of keratinocytes, and the cytoplasm is beginning to be filled with keratin filaments. So you can see here that they look a little darker in this illustration, and that's meant to represent these keratinocytes. This is also when the organelles begin to degrade. So essentially cell death is starting here. Once a cell becomes fully keratinized, they're dead, but they are very strong at that point. And you're going to find in this layer also extracellular lipids that help to form that epidermal water barrier. Then we can move up to the stratum lucidum. This is important. The stratum lucidum is not found in all of the epidermises across your body. The stratum lucidum is only going to be found in thick skin and the palms of your hands and the soles of your feet. This contains keratinized stratified squamous epithelium. It's thin and translucent. It's 2 to 3 layers thick. The cells completely lack organelles at this point, and so the main purpose is really going to be protection. And then last but not least is the stratum corneum. Stratum corneum is the most superficial layer. It also contains keratinized stratified squamous epithelial cells, as you can see from this picture how flat these cells are. But notice that the nuclei and organelles are even gone by this point. So it's comprised solely of these dead keratinocytes. This is the layer that is also constantly being removed because of different types of abrasion. So if you even take your nails and scratch your skin, you're pulling off some of these cells that are in the stratum corneum. So again, reviewing our little mnemonic: come, let's get some beer, corneum, lucidum, granulosum, spinosum, and basale. So this is showing some of the variations that you're going to find in the epidermis when you're looking at thick skin versus thin skin. So looking at this thick skin, you're going to find a very, very thick layer of the stratum corneum. This is the skin that you're going to find on the palms of your hands and the soles of your feet. And again, this is where you're going to find all 5 strata. You're going to find sweat glands here, but no hair follicles and no sebaceous glands that are going to be secreting the oil. This epidermis is going to be about 4 to 6 times thicker than what you're going to find in the thin skin. Thin skin, on the other hand, that you can see here on the right, covers most of your body. There is no stratum lucidum. So you can see we've got the stratum basale, spinosum, and then granulosum, and then we jump straight to the corneum. You can also see how much thinner that stratum corneum is. Here in this type of skin, you are going to find hair follicles, and you are going to find sebaceous glands and sweat glands as well. So what about skin color? Skin color is primarily determined by 3 different components: hemoglobin, melanin, and keratin. So hemoglobin, which you've probably heard of before, is an oxygen binding protein, and you're going to find this in red blood cells. So when it's bound with oxygen, the hemoglobin turns bright red, and this causes skin to flush or to blush. Melanin is a pigment that is produced and stored in the cells called melanocytes, and it can be transferred to keratinocytes in the stratum basale, which then migrate towards the stratum corneum. And so melanin, I should say, can occur in various forms that have either a yellow, reddish, tan, brown, or black shades. Now, interestingly, all people, regardless of the color of your skin, actually have the same number of melanocytes, but they differ in the melanocyte activity and the color of the melanin produced. and it's stimulated by UV light to make more melanin, or I should say UV light will stimulate these cells to make more melanin and to darken as well the melanin that's already present. And then finally, keratin. Keratin comes primarily from your diet, especially from yellow-orange vegetables such as carrots, corn, and squash. And it accumulates in the stratum corneum, but also in the subcutaneous fat. Keratin can be converted to vitamin A, which is needed for vision, and it also helps to improve your immune system. So that's our tour of the epidermis. What about now of the dermis? So the dermis is a thicker layer than the epidermis, and it's going to be found deep, and it's going to be made up of connective tissue proper. There are two major regions of the dermis. The papillary layer is the most superficial layer, and then the reticular layer is going to be below that. So the papillary layer consists of areolar connective tissue, and it has these dermal papillae that are connected to the epidermal ridges. This kind of looks like one of those egg crate foams that college students a lot of times will use on their dorm beds, for example. These little, those epidermal ridges are going to be, or I should say the structures themselves, the little finger-like projections, are formed at the deep end of the epidermis, and so then at the top part of the papillary layer is where you're going to find these dermal papillae. This helps to anchor the dermis to the epidermis. Within these dermal papillae, you can also see a few different structures here. You can find capillaries that help to supply blood and oxygen, especially to the epidermis. And then you can also find here depicted in this yellowish tone various types of sensory receptors, especially tactile receptors. Below that papillary layer is the reticular layer. The reticular layer is primarily composed of dense, irregular connective tissue. And you're going to find in this tissue collagen and elastic fibers that are in a mesh network type of pattern. In the reticular layer, you're going to find things such as hair follicles, sweat glands, sebaceous glands, nerves, blood vessels, muscular tissue. And this is an area where you're going to find essentially all types of tissue, because you will have even epithelial tissue that's lining some of these glands. When you have something such as, for example, stretch marks, this is being caused by tears that are in these collagen fibers in the dermis, and these can result from things such as excess weight gain or pregnancy. As I mentioned, you'll also find nervous tissue that provides innervation of the dermis. These nerve fibers in the dermis are responsible for monitoring sensory receptors in not only the dermis but also the epidermis, and they also help to control blood flow and gland secretion rates. You're also going to find tactile corpuscles and tactile cells that perceive touch sensations and communicate with other sensory nerve endings in the skin. Smooth muscle is found in the dermis, and you're going to find these in locations such as the erector pili, which are the muscles that are attached to the hair follicles and can cause the hair to stand up or to just lay in more of a neutral position. And then you're also going to find smooth muscle in the tunica media of the blood vessels that are in the dermis. So as far as that blood supply to the dermis, unlike the avascular epidermis, the dermis does contain blood vessels, and these blood vessels help to control body temperature. We mentioned vasoconstriction previously in this course, so that's the narrowing of blood vessels that help to preserve body core heat when the external environment is cold. And vasodilation is the widening of the blood vessels that assist in losing body heat and lowering the body temperature when the external environment is hot or when the body temperature begins to increase. And this makes the skin appear more flushed due to more circulation in the superficial layers. We mentioned previously that blood is a type of connective tissue that is composed of cells and cellular fragments. So the cells include erythrocytes, which are red blood cells that transport oxygen and carbon dioxide between the lungs and body tissues. The leukocytes, which are white blood cells that help to mount immune defenses. And then also platelets, which assist in blood clotting. The plasma of the blood is the extracellular matrix, and it's made up of protein fibers that are dissolved in a watery ground substance. And the plasma helps to transport nutrients, waste, and hormone. And there's also protein fibers in the plasma that come out of the solution to help coagulate or clot where vessels or tissue are damaged in order to help stop bleeding. So let's talk about some other features of the skin. How about tattoos? These are some great examples of anatomical themed tattoos. When you get a tattoo, if you get a tattoo, that occurs by ink being injected into the dermis itself. And that ink can then be trapped by fibroblasts, which, as we learned, are a type of cell in connective tissue. They're trapped by those fibroblasts and collagen, and so therefore that ink can last a lifetime. They help to prevent the breakdown of that ink. Now, something just to keep in mind, ink that's used in tattoos is not FDA regulated in any way. It contains polymers or vegetable dyes, but many types of tattoo ink can also contain things such as metal salts, including cadmium, iron, lead, etc. So that's something to keep in mind and to do a little research before you get something done of this sort. Now, what about this subcutaneous layer, or the hypodermis? Again, I really want to stress that this is not part of the integument. It is deep to the integument. We mentioned the hypodermis consists of areolar and adipose connective tissue. And because of that, primarily because of that adipose tissue, it serves as a shock absorber to the underlying tissue. It can store energy because fat has a metabolic use as well, and then it can also provide thermal insulation. The hypodermis does contain blood vessels, as you can see right here. And those blood vessels are why drugs that are injected into the subcutaneous layer can be absorbed faster due to their injection into the vascular system. And hair. Hair is formed by columns of keratinocytes, and you're going to find a hair follicle that surrounds each of the hair. In addition, I already mentioned this or alluded to it a bit, the erector pili muscles, which assist in straightening up those hairs. So those muscles can respond to emotions and cold temperatures by contracting, and when they stand up, that's actually what produces the goosebumps. Exocrine glands. 3 types of exocrine glands that you're going to find in the epidermis: merocrine sweat glands, apocrine sweat glands, and sebaceous glands. So the merocrine sweat glands secrete a solution that's about 99% water. And this secretion is clear, and it is controlled by the nervous system. You have numerous merocrine sweat glands on the palms of your hands, the soles of your feet, and the forehead, and it primarily assists in thermal regulation. Apocrine glands are simple coiled tubules that secrete their substances into the hair follicles, and you're going to find these in locations such as the armpits, the nipples, the groin and pubic regions, and also in the anus. Unlike the merocrine sweat glands, this secretion is thick and cloudy and has a lot of proteins and lipids. And it also causes bacterial growth, which gives it a distinct odor in these regions. And then finally, sebaceous glands. Sebaceous glands secrete an oil substance into the hair follicles or directly onto the skin surface. And this provides lubrication to help prevent the drying of the hair and skin. It's relatively inactive during childhood, but sex hormones that are released at puberty cause these secretions to increase significantly. So what about damage to the skin? Burns are classified by multiple degrees. If you see something that's labeled, or I should say, if something is considered either a 1st degree or a 2nd degree burn, those are considered to be partial thickness burns in terms of their damage that they're doing to the integument. So 1st degree burns involve only the epidermis, and these are characterized mostly by a redness and some slight pain. Sunburn is an example of a 1st degree burn, and this can just simply be treated by a cool, wet compress or cold water. Very rarely may need bandaging, but it has heals in about 3 to 5 days, and you're not going to find any type of scarring. 2nd degree burn, on the other hand, involves the epidermis, but also part of the dermis. So here's where the skin is going to start getting blistered and can turn into a reddish, tannish, or even whitish color. And this is the kind that tends to be definitely more painful. Just so you know, if this type of burn happens, you should make sure and take care not to break the blisters, so the risk, in order to minimize the risk of the infection. And also, do not apply any type of ointments, because those help your body to retain heat, and we want the exact opposite going on here. These tend to take longer to heal, usually more like 2 to 4 weeks, and can leave behind a slight scarring. And then there's the 3rd degree burn. 3rd degree burn involves not only the epidermis and dermis of the integument, but also the subcutaneous layer. And a lot of times these are destroyed completely. So regeneration will only occur from the edge due to the absence of the dermis. And so this is why skin grafting is often required to prevent any kind of abnormal connective tissue. When you have a third-degree burn, dehydration is a major concern because the entire portion of the skin has been lost, and so there's no way to help retain water across that area. You also have a much higher risk of infection because, again, the barrier is compromised, and so infectious agents can make their way into your body much more easily. And as a result, people with third-degree burns are usually given antibiotics to help prevent this. And last, I'd like to talk just a little bit about skin cancer. Most skin cancers are named based on what cell they actually damage. And now that you've learned more about these cells that you're going to find in the dermis and the epidermis, these terms may make a lot more sense to you now. So basal cell carcinoma, you now know what a basal cell is. And so this is going to be a cancer that initially begins by abnormal growth within these various basal cells. A squamous cell carcinoma is instead going to occur as a result of abnormal growth in the squamous cells of the epidermis. And then a melanoma is going to occur, again, starting with, as you may assume, with the melanocytes. So here you can see an example of these three different types. Squamous cell is going to be considered to be the least damaging and the easiest to remove. And at this point, you can see we're mainly damaging the epidermis, though it can go down into the dermis a bit. Basal cell, those are cells that are even deeper, so it's easier for them to invade the dermis. And then the melanocytes, it can be the worst type with these melanomas. And so you can see how this extends even deeper into the dermis and can also then invade some of the blood vessels, and then those cancerous cells can be carried to other parts of the body.